The host was Optimax. I'm here with the president of Optimax, Joe Spilman. Joe, how do you think the meeting went today?
Today was fantastic. We heard from a really diverse set of, companies that work in the freeform space in all sorts of different applications, from design to manufacturing to building systems and their applications. It was fantastic.
You could choose one learning point, one lesson learned, one takeaway. What would that be?
This is the freeform revolution. A lot of applications out there, and there's a lot more coming.
What would be the killer one? What do you think?
Oh, of course, Jose. Okay.
If I don't put you on the spot, then it's not me.
I learned more about what the CFO, Center for Freeform Optics, is doing at the University of Rochester, and their design tools are maturing rapidly and are becoming very useful for industry.
Welcome to Rochester, New York. I'm here with Susana Marcos, the director, David Williams Director of the Center for Visual Science of the University of Rochester. Susana, thank you very much for having us. What are we going to see today?
Thank you for coming, and it's always a pleasure to receive Optica here. So we're going to see a little bit of snapshot of the work that we do at the Center for Visual Science, which is a very multidisciplinary center at the University of Rochester. We have researchers from 9 different departments. We are in the facilities that are located in the medical center, and we're going to see some of the work that we do in the laboratories of visual optics, of retinal imaging. So it's part of the ARIA group, it's part of the Marcos lab, it's part of the Shalix lab.
This is a university hospital. You see, you have patients and you do research and you work together closely with the medical doctors.
Exactly. So CVS encompasses many different departments, but we are now located at the facilities of the medical center. We're very close to the Photonics Institute, which is the Department of Ophthalmology. And one of the advantages of this location is the proximity to patients, the fact that we can discuss with doctors, and the possibility to do this transfer of technologies that we develop in the lab to send to
Let's go.
patients.
How is the interaction with the eye doctors? Do you talk to them constantly? Do they join meetings? Do you have coffee breaks together?
Absolutely. And this is one of the advantages of the Center for Visual Science, that we bring people together with different interests around vision. And one of those, of course, is medical doctors. We host lots of colloquia, symposia, and we have projects together where we get their patients and we consistently discuss what is needed and how we can serve patients with the technologies that we develop.
For most of the companies developing technology for eye surgery, for eye inspection, clinical trials are always a challenge. Do you help with that? Do you have programs to help companies going through clinical trials?
We do, and there's different stages of clinical trials. My laboratory in particularly is more interested in the first stages, where we perform clinical studies on patients. We are not involved in phase 2 or phase 3 clinical trials, but yes, we work closely with companies in the eye space to first test their devices, first on human subjects using our visual simulators, and then eventually in the next phases.
Fantastic. Let's go meet your team.
Sure.
Saw each other quickly. And we are still at CVS, and we are meeting different parts of the center. I'm here with Jesse. Jesse, where are we right now?
Yeah, so we're in the Photonics Institute. This is the research center that is associated with the Department of Ophthalmology at the University of Rochester.
And what do you guys do here?
We do research on animal systems and on human research to improve ophthalmic care for humans that are undergoing treatments for eye disease, blinding eye disease, and also research understanding the mechanisms of what causes those eye diseases.
Fantastic. Are you going to show us some of the laboratories?
Yeah, do you want to come?
Of course, let's go.
Right.
And everything okay?
Everything's okay. It's just laser safety. We don't
Yeah.
have any lasers on in the room. Normally, we want protective eyewear, but
Yes.
we're safe today.
Very good. Let's go inside. So what
So
about
this is a very large optical table
Yes.
that comprises an adaptive optics scanning light ophthalmoscope, and this is actually a device that is quite famous and associated with the Rochester name. Some of the first adaptive optics ophthalmoscopes
Yeah.
were built right here in Rochester, and this is our latest innovation in these cameras.
Uh-huh.
The basic design is that we take a point of light,
Yeah.
a laser, and we scan it across the back of the retina. And unlike a traditional ophthalmoscope, which might fit on a small
Yeah.
benchtop breadboard, this is very large because it's a development instrument. It's a research grade instrument. Right here we have 3 different laser sources. Each one of these fibers that you see here contains a light of a different wavelength.
Yeah.
So we have visible spectrums
Yeah.
of light between 400 to 700 nanometers. We have the near infrared right here, and this is around 796 nanometers. And then we also have a slightly longer near infrared channel here. This is what we call our wavefront sensing beacon,
Yeah.
and that operates at around 904 nanometers. These two are invisible to humans. You can't really see that light, whereas the visible channel projects the rainbow of colors that we can see in our everyday life. So these 3 wavelengths, as you can see, they're separated, and one
Yeah.
of our first central goals is to make them coaxial. That's to combine all of the wavelengths into the same principal axis of the system. And they're all combined through these series of dichroics and flat
Yeah.
mirrors, so that we have these 3 separate lines and we combine them into one. Once they're collapsed into one, the light continues the same path through the system and ricochets off a number of telescopes. These telescope pairs allow that information to be projected into 2 planes. This is a retinal imaging plane and a pupil plane. The pupil plane is then transferred through a resonance scanner, which is right here, and a galvo scanner right here. And this takes a single point of light and it scans it across the back of the retina.
Excellent.
The light continues through the instrument and actually comes out all the way on the other side of the instrument. This instrument has been custom designed for the very tiny optics of a mouse eye. And the mouse is one of the most popular models for
Yes.
biomedical research. And the reason that we custom built this instrument is that we have a number of questions that we're trying to sort out in animal models before we bring that to human research. Once the light strikes the mouse eye, the mouse will be positioned on that side of the table.
Yes.
In a custom-made holder. The mouse is asleep during the imaging session, much like people would be asleep during general surgery. The mouse is asleep for about 2 hours, in which we have 2 hours of stable recording of their eye. The light, after it strikes the mouse's cornea, continues back to the retina, which
Yeah.
is the back of the eye. That's where our light-sensitive photoreceptors are, which gives us our sense of sight. And those are the cells that we're after studying in the eye. We would like to study the neurons,
Yes.
the glia, and in my laboratory to understand blood and blood cells inside the living eye. Once the light interacts with the eye, the light then comes out of the eye. It emerges back out of the pupil, and then it continues its path back through the instrument, through the same optical path that it went into the eye.
Yeah.
It returns through the same path, and then comes to this arm right here. This is what we call the detector arm. The detector arm is specialized because it uses something called a photomultiplying tube. It's a single point detector that basically gathers how much light came out of the mouse eye at a certain point of time. It then recompiles all that information to produce that image of that scan pattern of light. This instrument is really special because it contains an element that I neglected to mention, which is right here. This is a deformable mirror. And the deformable mirror is sort of the beating heart of the adaptive optics scanning light ophthalmoscope. It is kind of like a funhouse mirror. So when you bend and poke different actuators on this deformable membrane, it can design basically any shape, complex shape that you'd like. And the
Fantastic.
reason that that's important for ophthalmoscopy is that the front of your eye blurs the image of the back of the eye, and that deformable mirror allows you to bend and correct the light to compensate for those miscalibrations, misalignments, and bumps and imperfections in the front of the eye. Once those are corrected, now you've got the sharpest image that you can get of the back of the eye.
That's a commercially available, the form of mirror, correct?
Yeah, so this is actually from Alpeo.
Yes.
And so this is a very common deformable mirror that's used in a lot of ophthalmological research.
Fantastic. Let's say hello to Alpeo, Optica Corporate member. What else are you going to show us today, Jesse?
Yeah, so we're going to show you a number of different things in the laboratory. One is basically just the fundamental design of these large optical tables. Here at the University of Rochester, we have a large number of these instruments. This is one, but we actually have a total of 6, and actually working on the 7th adaptive optics instrument here, all within the laboratory of the same laboratories. We have some that are dedicated for different species. So we have some for humans, some for small animal research, and other laboratory animals as well. Why do we have so many? We have so many because each one has a different performance specification. Some are optimized for fluorescence detection. Some are optimized for 2-photon imaging, so looking at the nonlinear interactions of photons of light. And then some are custom built, again, for very tiny mouse eyes. When you look at the front of a human eye, the pupil acts as sort of the aperture, or the numerical aperture of what dictates how much light and how much angle you can put into the human eye. In a mouse, we have a very, very tiny aperture. It's only about 2 millimeters in diameter. So this instrument has been custom built for the mouse eye because it has a smaller numerical aperture. And you might think that resolution would suffer, knowing the fundamentals of the Abbey limit, but there's something very special about the mouse. Even though it has a tiny aperture, it has a very short axial length. And what that allows you to do is it increases the numerical aperture and actually boosts the amount of resolution that you can get in these very tiny eyes. So there's a few, in the commercial domain, there's a few exciting things that we're working on. One is that a lot of the products and specialized optics that we're using in these instruments, a lot of them are actually built, designed, and manufactured right here in Rochester, New York. So this is a really cool place to be doing this work because we have commercial partners that are right here. But as you pointed out in the instrument, also a lot of very important global partners in producing very specialized optics that allow this kind of research to be done in the research domain. In terms of getting this instrument ready for the commercial, a commercial ophthalmoscope, there's many groups that are interested in doing this. The technology has been around for, at least in the eye, for around maybe 25 to 27 years. And there are many groups that are trying to get this miniaturized, right? So getting the optics toleranced for a very small platform where you can wheel this in room to room. And there's also a lot of study in terms of what the clinical utility will be. When you talk to ophthalmic doctors, not all of them want the same information from the eye. Some of them care about the best possible resolution. Some of them care about specific imaging modalities like fluorescence, how much fluorescence is coming back out of the eye. Some of them care very much about vascular perfusion, how much dye, when they inject into the vein of a human, how much of that dye is getting into the eye, and what does that tell you about the health of the blood flow in the back of the eye. Others are looking at these incredible phenomenon like autofluorescence. In the back of your eye, there are proteins that and aggregates that naturally fluoresce. And other clinicians will use that as a biomarker for, again, the health of the eye and how far a disease has progressed in a human subject. I think one of the untapped commercial potentials for these devices is not only with optics industries, but one thing that gets me very excited is that we, as a group in Rochester, try to partner with pharmaceutical companies to try to help them narrow their focus on which drugs and which drug targets have the best chance of success. And we feel we have an advantage in that because you can image individual cells, and with that microscopic resolution, you can see how cells survive or how they may die, how they may interact with each other over the course of disease. That's a very important thing for pharmaceutical drug companies to look at, because as they're developing the drug, they don't just want to help the end goal of the patient see better, but they want to understand what the mechanism is that's causing the disease so that they can better tailor their therapy. So one of our interesting and motivated partners in the future, I think, will be working with not only industry, but pharma as well, so that we can use this technology in the most instructed way to help the most people that we can. So in Department of Ophthalmology, we have a number of clinical faculty, and we have dedicated research faculty as well. I believe the current census is around 14 faculty that do basic science research. I'm the director of a group called ARIA, which stands for the Advanced Retinal Imaging Alliance. And that is a collection of 5 laboratories that all use this adaptive optics technology, each laboratory with a different goal. In that group, there are over 45 associated people using this adaptive optics technology. And the cool part about the distribution of those people is it ranges from undergraduate students that are working on their optics degree and trying to train about how you understand and build these complicated optical devices, to graduate students that are specialized in the Institute of Optics, also biomedical engineering, neuroscience, pathology. All of these students are interested in building these technologies either for science questions or for engineering questions. And then we also train medical residents and fellows, postdoctoral fellows. And then we have a huge and valuable part of our team is the technical staff that makes this work possible. Talented optical engineers, software engineers, animal technicians, human imaging specialists, and of course the faculty. So all told, that's about 45 people that make these laboratories possible.
All right, let's see the next one.
Yes, let's go in here.
Biosafety level 2.
Yes.
What does that mean?
So this is biosafety level 2 because a lot of these laboratories are doing animal research. Some of the animal research that we're doing uses very specialized proteins to express, for example, fluorophores or animal models that replicate human disease. That makes special considerations for biosafety level 2. We have to have the appropriate accreditations and training for all of our staff that work on biosafety level 2. So we're going to go out of this very tight room.
Yes.
I'll leave this open.
No one here, so we are.
So this is the second of those 7 laboratories that I told you. Each one of these adaptive optics cameras takes up a separate room. As you can see, the footprint is pretty large. These tables are around 6 feet by 4 feet each. And the fundamental design behind each one is very similar. It has, for example, the deformable mirror that I talked about earlier. It has something called a wavefront sensor, a Hartmann-Shack wavefront sensor, which is the way that you measure the imperfections of the front of the eye. Once you have
commercially available Hartmann-Shack
sensor? So we work with a number of different vendors for the Hartmann-Shack wavefront sensor. For these, we've combined with a number of cameras, CCD, scientific grade CMOS or CCD cameras, and that's combined with a lenslet array. From a number of different vendors, each with a different performance specification of the focal length of that lenslet array and the array patterning of those lenses.
Sensors, we know Imagine Optic provides one. Who else do you have?
I'm not sure if this one would go into the production, but we've worked with government contractors as well to produce these wavefront sensors. Again, each one is sort of custom built for the design specification of the instrument that we're using.
We also had a nice meeting yesterday on Freeform Optics here, and we saw PHASICs as well.
Yes.
Wavefront sensors for characterization of optics. It's great that you're working with these companies.
So, I mean, I think in your role, Jose, you're probably looking for, what some of the bigger name vendors are that we use. And I'm happy to sort of name and call out some of the vendors that we use more commonly. So in these instruments, we use everything from very high power and precise light sources. We've had wonderful collaborations with spectrophysics for 2-photon light sources. We also have a close partner just outside of Rochester, New York, in Fairport through Optica, which uses multispectral lines. This gives us our visible light in what they call light engines, which allow us to very flexibly put in different colors of light into
What are the most interesting wavelengths that you normally need?
For our purposes, our most common one is 488 nanometers. And the reason that that's a very common one in biomedical research is that there are a lot of fluorescent proteins
Yes.
that are being designed for maximum excitation at 488 nanometers and then fluoresce around 520 nanometers. However, we also use lines at 515, 568, and 640 nanometers. Each one of those has a utility of a different fluorophore, a different fluorescent protein.
There's a narrow line width or high power. Do you have any extra demands beyond what is commercially available?
Yeah, so I think our biggest demand for these light sources is the stability of the light source. And stability can be measured in a number of different ways, but we ideally like polarization maintaining light sources and power stability. So when we're imaging with either a human or an animal subject, we want a user interface that is very precise, repeatable, and user intuitive when you interact with the light source. Because yes, these high performance lasers, you can take a while to train on them and become an expert. But if you have it so that even a novice user knows how to interact with the user interface, that's probably the biggest headache that we deal with. Optica has built a wonderful intuitive interface that we like very much. The other, I think, is again just the stability. The price point is of course a big considerations for research laboratories. When we scale the number of these in the laboratory, we have 7 of them. But when they go out into the commercial domain, getting a stable and inexpensive light solution will be critical for pushing this into the commercial domain.
Fantastic. Walk us through the setup. What do we have here?
So in this system, we have a number of different light sources. Actually, on the table right here, this is a light source that comes from Superlume. Again, a very stable near-infrared light source that's
It's a
fiber coupled.
superluminescent diode,
It is.
all the way from Cork, Ireland.
That's correct. And so this broadlighter has been sort of a staple in our laboratory for a long time. This device has provided SLD powers in the correct power regime that we need. In the eye, unlike other devices, sometimes light output is in hundreds of watts of power. In the eye, we're usually down into hundreds of microwatts. Hundreds of microwatts is sort of the limit for us for these light devices because the eye is, of course, very sensitive to light. We can only put in so much light into the eye.
Yeah.
But getting that light very stable is critical for us. That superlume is then piped into the instrument through the fiber coupled cages that you see here. And each one of these cages allows the light to be combined, just as we had in the other instrument, and combined into the path in a coaxial way. Each one of the lights, the visible, the near-infrared, and the wavefront sensing beacon, ricochet through a number of these telescope pairs. Like the other instrument we saw, scans the beam across the back of the eye, and then eventually comes out through the opposite side of the instrument. In the opposite side of the instrument, that little point of light is scanned in through the subject's eye and creates a rectangle. We call this the raster pattern. That light then, when it interacts with the neurons, the glia, and the blood cells, contains information. It contains spectral information, it contains phase information, it contains intensity information, and all of this different properties of light that we can measure. And then the light comes back out, emits out of the eye, and is detected by our photomultiplying tubes.
Yeah.
We have been using Hamamatsu photomultiplying tubes for many years. Again, a price point that is amenable to scientific research, a sensitivity that is really exquisite, and a stability in our hands that has had a shelf life, usually for about 3 to 4 years for these detectors. One of the things that we would love in these detectors is a little bit more longevity. Having a detector that can withstand overburdening it with a barrage of light, overexposing a PMT is a really challenging thing that degrades the lifetime of these instruments. Getting those PMTs to be lower cost and not as sensitive to overload would be a fantastic thing in the future.
Photomultipliers are fantastic detectors. They've been in the industry for many, many, many years. Have you tried any of the upcoming new ways of detecting this kind of wavelengths?
Yes. So you're talking about our next generation of these instruments. We're very excited about continuing with the photomultiplying tube solution and APDs, avalanche photodiodes, but also very excited about silicon-based detectors and array detectors. So things like SPIMs and SPADs
Yes.
are really exciting detectors because now they're starting to hit quantum efficiencies that rival PMTs. They have a robustness that PMTs have not traditionally had. And then also the fact that you can get very fast readout from array detectors is now starting to overcome the limitations that those devices have had in the past.
And the arrays are becoming larger and larger, and the price is actually being reduced every
coming
year.
down,
So
yes.
this is
We're very, very excited about that. And then just in the last 2 minutes or so, Jose, like the vendors that we're using quite often here in these systems are some that are common to many laboratories, but we do a lot of our sourcing through Thor Labs, of course.
I can see that. Yes.
Fantastic partner over the years for building and prototyping these instruments. They're very fast in terms of their off-the-shelf components, the diversity of products they have, and again, price point that is amenable to research devices. We also have a lot of, for example, filters through Semrock
Yeah.
and Chroma. Those have been fantastic partners both regionally and locally to use these devices, which have very strict tolerancing for the wavelengths, for example, that they pass. And then we've also partnered with companies like Lambda to create custom optics
Of course.
with achromats and doublets that have a very unique specification for these ophthalmic
What would be
devices.
your number one in the Santa Claus Christmas list to the photonic industry? What is the kind of device that you would say, oh, wow, this could be fantastic for my setups?
Yeah, so I think our most exciting technology is the array detection.
Yeah.
So in these devices, we have done almost everything that we can to project a very tiny point of light into the retina. And as I described to my students, that's really where the photon adventure begins. The return path in which that photon comes out and must be detected. I think that's the biggest gains that we're going to see in the years ahead is in the detection, the efficient detection of those photons, which are very rare and precious, especially in ophthalmic imaging. In these instruments, we have a number of different biomarkers. We're looking at blood flow as one of the most exciting biomarkers in these devices. Blood flow is not only exciting for imaging in the eye, but also for evaluating systemic disease. The eye just happens to be this clear window where you can peer into the body and get a very clear picture of not only the red stuff that pumps through your arteries and veins, but actually individual blood cells that move through the tiniest of vessels through the back of the eye. So blood is, in my opinion, one of the most important biomarkers that one can have to diagnose human health. And here we get access to it in the eye. This is Jin He. He is an optics student.
Hello, Jim. Nice to meet you. What do you do?
My current project is developing an in vivo flow cytometer for the mouse retina.
An in vivo flow cytometer.
Yes.
And you do it here?
Yes.
Can you show it to us?
So we have two detector channels, two separate PMTs optimized for two different wavelengths. So we have a near infrared PMT on the right that collects the information about the amount of blood that flows through a certain vessel. And we have a fluorescence channel which detects the fluorescently labeled blood cells. And we can count the number of cells and at the same time recording the amount of blood that's flowing through this vessel so that we can get a population estimate of how many cells are present in a given volume.
Hello.
So welcome.
You should know I always come with a camera. Yes. So, Susana, so where are we right now?
So we are in front of the Adaptive Optics Visual Simulator. So this is a system where normally a patient sits in it, and we project. So we can manipulate the optics of the person.
I already see a super continuous
So you
generation
see a
source from
super
Lucas
yeah,
using the
exactly.
fiber from Lever Flore.
Yeah, so we have optotunable filters, we have optotunable lenses, we have adaptive optics mirrors, we have...
Are the adaptive optics commercially available?
Yeah, so this is from Imaginize. We have a spatial light modulators. And basically what we do here is we have a person experiencing the world through manipulated optics.
Okay.
So with a Harman Schaap wavefront sensor,
Yes.
we can measure the aberrations.
From Imaginize.
Yes. We can measure the aberrations and we can correct that with a deformable mirror so that the person can see the world without no aberrations. And then with a spatial light modulator, we can model different intraocular lenses, different contact lenses so that we can perform visual testing, visual psychophysics,
The
explore
special light
visual
modulator
perception.
is from HoloEye.
It's from Hamamatsu.
Hamamatsu. And from HoloEye, we have on top the diffractive optical element. Okay. Wow, this is great. It's like having all our corporate members working together.
Yeah, and so what we do is basically work on new designs of contact lenses, new designs of intraocular lenses for presbyopia and myopia. Myopia affects 90% of the population in certain parts of the world. Presbyopia affects everyone over a certain age. Cataract surgery is the most performed surgery in the world. So basically what we can do here is to perform these surgeries virtually. With the patient experiencing the world through those designs. So we work with develop new designs. We work with industry that comes up with new designs, and before
Before doing the
launching them-
surgery, they come and they sit down, and this is how you're gonna see after.
Yes, this is exactly how it works. So, these, of course, before implanting the lens, but also before manufacturing the lens.
Yes.
So this is the streamline the process of selecting.
Not only the patient is experiencing what's gonna happen after, but knows exactly which design is gonna need.
Yeah, for the same patient can experience different designs. The selection is personalized. This we have actually brought to an instrument that is see-through. Is something that the surgeons have in their clinics.
Yes.
But this is like an experimental prototype that is very versatile, and we can produce very easily any lens that you can imagine.
Why didn't you introduce us to your team?
Yeah, so this is Sabia Waswami.
Hi,
Sabia is
Thanks. So basically, I am working on simulating different lanes that essentially we can use for myopia control, right? As Dr. Marcos mentioned, myopia is going to be a boom, right? And then we are having different factors that can trigger the axial elongations of myopic eyes. And then here we are having different lanes that can correct myopia and also that can stop the eye from going more. And we are trying to understand which lanes can perform better. As we can simulate different optics of the lens and we can do psychophysics and we can understand which lens is actually give us better visual functions. If we can do that, then we can suggest, okay, you can use this lens for the patients and this patient is perform better.
Xavier, by the way, is a student in brain and cognitive sciences, and Tian Lun is a graduate student in optics. So you can have an intro.
Yeah, hi, I'm Tian Lun, and so the first day I joined the lab, here was only an optical table, and I was building the system with the engineer Katik, and then we spent almost 3 months to have the first version of the system, and then we fine-tuned with Sarah for a long time, and we keep upgrading the whole system. And it's very good to start with scratch, building a system, because you know everything. When you move something, you know where might have some like feedback, so you know how to fix everything. And for this whole system, the most impressive part for me is I was born with very, very high astigmatism. So the first day that I put my eye into the system, I see very clear, very sharp image that surprised me.
Thank you very much for this. That was great to meet you, and we're gonna have a coffee later, correct?
Yes.
See you later. Bye bye. Thanks a lot. I love that part when you see the PhD students so excited about their job.
Thank you.
So
you have more.
inspired about their
We
job.
have more. Tenemos más, pero yo creo que
No hay tiempo, esto es
siempre.
precioso. Me ha encantado y tengo hasta goosebumps. I have goosebumps.